Hello, welcome to the biology class. In the previous class, we have discussed a brief idea about the plant breeding. Okay, we just have understood what is plant breeding and how the plant breeding has came into practice, and we just have mentioned the steps in plant breeding. Okay, we didn't see all those steps in detail, we just have mentioned those steps. Okay, in today's class we shall see those steps in detail. Okay, how that process will be done and what is the use of that step, etc. Okay, so looking at the first step of the plant breeding, that is the process of collection of variability. Collection of variability. Look at the step, look at the word variability. Variability refers to different plants of the same species, different crops. If I am taking, for example, wheat, collecting the wheat seeds from different areas. One area where the wheat is grown very well, from that farmer collect some seeds of the wheat. Other area collect some seeds of the wheat from other farmer. Like that, visiting different places and collecting the better yielding varieties, better yielding crop varieties of one crop, say wheat, say jowar, say rice, etc. Okay, so variability refers to collecting various types of genes, hence we use the word germ plasm collection here. Germ plasm collection means it is also called germ plasm collection. Then what is germ plasm here? Here, whatever the living material, what we collect for growing the next seed, next generation of the plant, what is what we mean by germ plasm. For example, I have preserved 1 quintal of jowar as a seed for next generation. I have preserved 1 ton of sugarcane for next season. So whatever seeds, whatever plant materials we preserve for next generation, for growing the next generation of the crops, that plant material preserved is called germplasm. Okay, germplasm here, it may be seed. Germplasm, it may be plant cutting. It may be fruit. Okay, whatever the living material, whatever the plant material we have preserved for growing the next season, that is what we call germplasm. Okay, so here we collect the wild varieties, we collect different species, and we also collect the relatives of cultivated plants. We also collect relatives of the cultivated plants. Simply collecting different source from different sources. Same plant of the same genus species, but growing in different areas. Okay, that is what we mean by germplasm collection or collection of the variability. Okay, so simply visiting different places and collecting the better yielding varieties of the plant. That is the first step here. Remember the word germplasm. Okay, it is any plant material preserved or collected for growing the plant in next generation. Better we shall write germplasm is nothing but any plant material preserved or collected for growing that plant in next generation. Is called germplasm. Okay, so simply it is the plant material. Material may be seed, cutting of the plant or a fruit, etc. There may be one more question on this germplasm. Right? Better you once write down the definition and we shall move further. After collection, when you visit different places, when you collect different varieties of the plant, what will be done after bringing all those steps? Okay, then second step will be evaluation and selection of parents. I use the word parents here, right? Parents means the one which give rise to next offsprings. One plant produce many plant, one animal produce many animals. So the first animal is called parent here, the first plant parent plant. Okay, means for the process of reproduction we are selecting the parents. For the process of pollination, fertilization, seed formation, we are selecting the parents. But before selection we need to evaluate. Right? Means whatever the plants we have collected here, among them we must perform screening. Screening means pick up the good one and discard the bad ones. Means among the 10 plants you collected, say 6 plants were found better, then pick them up, take it for next generation. Among them 3 are found not that satisfactory, discard them. That's what we call screening. Means selecting the better one, selecting the good one among how much we have collected. Okay, after evaluation then we should select some plants as male, some plants as female, because plant breeding itself is the pollination performed under human control. Okay, the process of pollination, cross pollination, which is performed under human control. Okay, so the pollination is performed under human control. How it is done we will understand in the next step. Okay, but in this step we just are comparing those ones what we have collected. Among them we are selecting good ones, we are preserving good ones and removing the bad ones. Okay, ಯಾವುದು ಚೆನ್ನಾಗಿದೆ ಅದನ್ನು ಇಟ್ಕೋಬೇಕು, ಯಾವುದು ಚೆನ್ನಾಗಿಲ್ಲ ಅದನ್ನು ತೆಗೆದು ನೆಕ್ಸ್ಟ್ ಸ್ಟೆಪ್ಪಿಗೆ ರೆಡಿ ಮಾಡ್ಕೊಳ್ತಾರೆ. ಓಕೆ, ಥರ್ಡ್ ಒನ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ದಟ್ ಇಸ್ ಅ ಮೇನ್ ಸ್ಟೆಪ್ ಆಫ್ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್, ದಟ್ ಈಸ್ ಕ್ರಾಸ್ ಹೈಬ್ರಿಡೈಜೇಷನ್ among the selected parents. Cross hybridization, nothing but cross pollination. Nothing but cross pollination means here they have to select some plants. They have to select some plants as male. They have to select some plants as female. They have to select some plants as a female. So you all know that pollination is a process of transfer of a pollen grain from anther to the stigma of different flower of different plant. Different flower of a different plant. That is what we use the word pollination. Right. Here we are performing the pollination by manual pollination. That is what we use the word cross hybridization. For that there is a procedure with some 4-5 steps. Okay, in that first step is emasculation. Hope you remember in the second chapter, that is sexual reproduction in flowering plants, there is a study for you, that is artificial hybridization. Once you recall this from the second chapter, the same thing will be done in this step. Whatever the steps you have followed in this process, what you discussed in second chapter, in this title, in this concept, the same thing is done here. Okay, so here in this step, the first step is emasculation. Look at the word masculation, male gender. E, removal of the male gender, that is removal of anther. Removal of anther from bisexual flower. Bisexual flower means a flower having androecium and gynoecium, male reproductive part as well as a female reproductive part. So from the bisexual flower having androecium gynoecium, here they are removing the anther. They will remove the anther. Okay, so the word emasculation, removal of the male gender. Okay, look at this. E refers to removal, like masculation, masculine gender, male gender. Okay, simply from the flower anthers are removed. Okay, in the plant, in the nature where they are grown, maybe in the greenhouse, maybe in the forest, from that flower the anthers are removed. Right. Second step, bagging. Second step is a bagging. Means the flower after removing the anther will be covered. Better I will just draw a small flower diagram, then you understand better. Say this is the plant. This is the plant having the anthers. This is the flower. From this flower, first step, emasculation, removal of the anthers. Removal of the anthers. So now the flower has only gynoecium, only female part of the flower. The second step is bagging. Bagging means the flower will be completely covered. Flower will be completely covered by using any cloth or the polythene bag. Okay, why they have to cover? Because there is a process called pollination, right? If you keep the flower open, the pollen of any species may fall on the stigma. ಫ್ಲವರ್ನ ಹಾಗೆ ಬಿಟ್ಟರೆ ಫ್ಲವರ್ ಮೇಲೆ ಯಾವ ಪ್ಲ್ಯಾಂಟ್ನ ಪಾಲನ್ ಬಂದು ಕೂಡ ಬೀಳಬಹುದು. ಓಕೆ, ಅವಾಗೇನಾಗುತ್ತೆ, ನೀವು ಫ್ಲವರ್ನ ಕ್ಲೋಸ್ ಮಾಡಬೇಕಾಗಿರೋದಕ್ಕೆ ನೀವು ಸೆಲೆಕ್ಟ್ ಮಾಡಿರೋ ಪ್ಲ್ಯಾಂಟ್ನಿಂದ ಪಾಲನ್ ತೊಗೊಂಬಂದು ಡಸ್ಟ್ ಮಾಡಬೇಕು. ಆದರೆ ನೀವು ಅದನ್ನು ಓಪನ್ ಆಗಿಬಿಟ್ಟಿದ್ರಿಂದ ಏನಾಗ್ತದೆ, ನೇಚರ್ನಿಂದ, ಫಾರೆಸ್ಟ್ನಿಂದ, ವೈಲ್ಡ್ ಪ್ಲ್ಯಾಂಟ್ಸ್ನಿಂದ ಅಥವಾ ಅದೇ ಸ್ಪೀಸೀಸ್ನ ಬೇರೆ ಪ್ಲ್ಯಾಂಟ್ನಿಂದ ಪಾಲನ್ ಬಂದು ಇದ್ರ ಮೇಲೆ ಬೀಳಬಹುದು, ರೈಟ್. ಸೊ ದೇರ್ ಮೇ ಬಿ ಪ್ರೊಸೆಸ್ ಆಫ್ ಕಂಟ್ಯಾಮಿನೇಷನ್. ಓಕೆ, so to avoid the contamination of the pollen from other plant, we need to cover the plant, cover the flower, cover the emasculated flower with a polythene bag or etc. any cover, right. Then when the flower is covered, when the flower is bagged, after some days, when in other plant, when in other plant, other parent, when the anthers are ready, anthers are ready, then once remove the cover, once remove the cover, take the pollen, dust on the stigma, dust on the stigma, and again cover the bag on the flower. Okay, this step here we call it as a cross pollination and rebagging. And rebagging means take out the bag, dust the pollen and add the bag again, cover the bag again, cover the flower again. Okay, so cross pollination, dusting the pollen on the stigma, bagging, rebagging means covering the flower again. Okay, here the cross pollination is done with desired pollen, desired pollen. That is the third main step. Fourth step, fruit formation and seed setting. Of course, we should not do anything, just leave the plant. After pollination, after rebagging, don't touch the plant. Naturally, the plant undergoes pollination, fertilization, formation of the fruit, formation of the seed, etc. Okay, so when the fruit seeds are formed, the last step will be collection and preservation. Means either the fruit is collected or the seed is collected. Okay, that's how the pollination is performed manually, man-made pollination, artificial hybridization. So we all know that anything made by human is artificial. Anything made by human is artificial. If anything available in nature, it is natural. Right, natural, artificial. If anything is done by us, it is called artificial. So here the hybridization, the pollination is done by human beings, hence it is artificial hybridization or cross hybridization. Okay, so remember, when we cross two plants, when we cross two plants, means plant A having some characters, plant B having some characters. When both are mixed, when both are crossed, some of the character of plant A, some of the character of plant B will be observed in the progeny plant C. For example, this character, this character, this character, this character, this character, this character, they all may be present in this plant, and this plant will be better than these two. C is better than A and B, means the offspring, the progeny plant will be better than both the plants. Okay, that's why this plant, this generation is called hybrid. Okay, this is called hybrid. So the name hybridization means formation of the hybrid is hybridization. Cross by crossing the two plant species. Okay, same species, two different plants. Right, that is the third main step of the plant breeding. So here remember these 5 steps in this step. Okay, how the cross hybridization is done in the plants. 4th step here, testing and evaluation of the superior recombinants. Selection and testing of superior recombinants. What is this? Superior recombinant, a new word has come. I said earlier, we have crossed plant A, we have crossed plant B, we obtained the hybrid as plant C, right? Similarly, we might have crossed A1, B1, the offspring C1, right? So, among these hybrids, say I got many such hybrids. I got some 10 hybrids. 10 hybrids. Among 10, if you found some 5 better than the other 5, then those are called superior recombinants. Okay, I have 1, I have 2, 3, 4, 5, 6, 7, 8, 9, 10. Among how much you have obtained, you can compare. These are better, these are less compatible. So you can select those better ones. Those better ones are called superior recombinants. Okay, and for those superior recombinants, you need to perform one process, that is selfing. Selfing to form pure lines. Selfing means if the plant is bisexual, then don't touch the plant, leave it. It will produce the flower. The flower has androecium, gynoecium, anther, stigma. Then in the flower itself, the pollination takes place. Okay, so a new plant will be formed. Again, next generation, don't touch the plant. Plant grows, flower is formed, pollination occurs. If you do the same for 4 to 5 generation, then the progeny will be known as a pure line, since it has high level of homogeneous characters. Homogeneity means all the characters are same as that of the parents. When there is cross pollination, there may be mixing of character, but when there is a self pollination, there will be no mixing of the character. Hence, the plants will be having homogeneous. Okay, so it is also called inbreeding. Inbreeding. Okay, it's all done by self pollination, just by letting the plant to undergo cross pollination or etc. Right. So just select how many are there, how many are better, then how to test them. Testing means growing in the plant, growing in the field. Selection is choosing the better one. Testing is nothing but growing the plant in the field, allowing it to self-pollinate, allowing it to form the fruit, seed, looking at the new crop. Again, repeat the same, cross-pollination, self-pollination and fruit seed formation. That is what we call testing. Right. Simply here, recombinant means the offspring of 2 parents, the offspring of the sexually reproduced plants. And the last step here is very important, testing, release and commercialization of new cultivar. or new variety. New cultivar also known as a new variety. Okay, means by following all these steps, one high yielding variety is found, then that high yielding variety will be selected as a new cultivar, selected as a new variety, then that variety should be brought to the market. Means all the farmers should know about that variety, all the farmers should purchase the seed of that variety, all the farmers should grow that seed in their field, that seed must be grown in the farmer's field. Hence it must be commercialized, means it must be brought into the market, it must be brought into the market. But for that testing is very important, testing is very important. Then how this testing is done, right? You should know how the testing is done. Means what are the criteria, what characters are observed during the testing, right? So we all know that for the main aim of the gradient evolution is to increase the yield, increase the yield. So in terms of yield, they will check the amount of yield, means how much seeds are produced in this year, how much seeds are produced in 2 years, like that amount is calculated, amount is measured. Then quality of yield, means among this 10 kg of the seed produced, how much kgs are better for eating, how much is good for eating. The seed of this crop is good, seed of that crop is not able to eat, like that they will check about the quality of the yield. Then duration of exposure to nature, to the climatic conditions, means unfavorable condition, means how long the plant can tolerate, how long the plant can tolerate. And resistance, resistance to diseases and pests, diseases and pests. So if the plant has all these properties, then that plant will be brought into the market as a new variety, that plant will be brought into the market as a new cultivar, so that the farmers can buy those seeds or those germplasm or those seedlings if it is any one of the horticultural crops, right? That is what the criteria expected in the new cultivar developed by these 5 steps, right? And these will be observed under the conditions like, under the conditions like normal fertilizer application, normal fertilizer application, under normal irrigation, and normal agricultural practice. Means whenever all the agricultural practices are normal, water, fertilizer, under the normal condition, if these are the characters present in the plant, then it will be released as a new variety. If any one of these is missing, okay, if the amount is good, less quality, plant will not be accepted. Okay, if the quality is good, amount is less, then the plant will not be accepted. If both are good but it cannot tolerate the temperature, can't tolerate the humidity, then again plant will not be accepted. Like that, if the plant has to fulfill all these conditions under this criteria and at this available facilities, then only the plant will be released as a new cultivar. Okay, and also testing is done in various regions. Testing is done in various agroclimatic regions. Various the word agroclimatic means different climate for agriculture. Rainy area, not suitable for agriculture. Dry area, somewhat good for agriculture. Medium rain, best for agriculture. Like that, we have many agroclimatic regions in India. Okay, some area is getting more rain. We can't grow the normal small croplands in that area. Like that. Okay, and also it is done in 3 to 4 farmers' field. Means the same crop, cotton plant, cotton grown in this farmer's field, cotton grown in this field, cotton grown in this field. Then they will compare, look at this crop, this soil, look at this soil, look at this soil, how the plant has grown. Also, they will compare with local varieties. They will compare this plant with local varieties of that area. For example, I have developed one variety of seed in Jamakandi, but I want this seed to be purchased by the people of Mudhol, etc. So I will test this seed in Mudhol also. I also want the plant to be purchased by the farmers of the Bagalkot. I will also test the seed in Bagalkot. Also, I want the seed to be purchased by the farmers of the Bijapur. So I will test the plant in Bijapur also. So means different places having different climatic conditions, agroclimatic conditions. So when this seed developed in Jamkhandi grows well in Bijapur, yes, they can sell that seed in Bijapur. If the plant grows well in Mudhol, yes, they can sell that seed in Mudhol also. If the plant is unable to grow in the soil of Bagalkot, then people will not purchase that seed. So this will not be possible. Like that, whatever the plant developed in one area, that will be tested in different agroclimatic regions. Okay, wherever it is not possible to grow, then in those areas the marketing of that seed, that variety, that cultivar is difficult for that company or that team or that research institute. Okay, that's how the plant breeding will be practiced in these 5 steps. In theory examination, it is the most commonly asked 5-mark question. Most commonly asked 5-mark question in this chapter, that is strategies for enhancement in food production. Okay, so first remember all the 5 steps and try to explain what is the meaning of that step. Okay, no need to write complete details, because if you go on writing, it may reach 4 pages, right? So don't try to write all the things in theory examination. Just write meaning of that line. What does it mean? Okay, that's enough. Okay, so these are the basic steps of a plant breeding, right? We'll now see some of the high yielding varieties of crops in India, and those plants which are used by the common man or the industries regularly. Okay, mainly the high yielding varieties of India. Okay. High yielding varieties of crops in India. Okay, high yielding varieties means the crops which give more yield, more profit. India is basically an agriculturally dependent country. So agriculture is one of the major sources of income for the farmers. Okay, so here India contributes, means the agriculture contributes about 33% of GDP to the India. Gross domestic production, GDP. Okay, agriculture contributes for about 33% of the GDP, and the agriculture employs about 62% of Indian population. Means among the total population, 62% are dependent on agriculture or agriculture related business. Okay, whatever topics we discussed in earlier classes, like fishery, like poultry, apiculture, so they all come in agriculture only. Okay, all coming under agriculture. So about 62% of the people of India are dependent on agriculture. Okay, but what about the agricultural land? Agricultural land, it is the same. Agricultural land is the same. It is not increasing. Agricultural land is not increasing. Right. So with the same agricultural land, we need to increase the yield. Okay, so now the focus is production per unit area. This must be increased. Okay, our focus must be on increasing the production of the crop per unit area, means how much area available. In that area, the production must be increased because the population is increasing as usual. Okay, to feed the increasing population, the production per unit area must be increased. Okay, for that we have the high yielding crop varieties like wheat. We have high yielding crop varieties like rice, we have sugar cane, and we have some of the millets, etc. Today we shall see about the wheat, then we will see rice in the next class. Okay, so first we shall take high yielding varieties of wheat. So first one, so you all know that wheat is one of the major food crops in India. Okay, so we all know that chapati and many more food items are prepared, some biscuits and many more foodstuffs are prepared from wheat. Hence, it is one of the major food crops in India. Okay, so we all know that about 1960s the production of wheat was just about 11 million tons in India. In India, the production of wheat was about 11 million tons in India. But by the end of year 2000, the production has been increased to 75 million tons. The production is increased to 75 million tons. What is the drastic change? 11 million tons to 75 million tons. Okay, so this drastic change in the production of wheat is because of green revolution. Green revolution by Nobel laureate Norman Borlaug, who at the Mexico has developed some of the semi-dwarf varieties of wheat. In Mexico, he has developed some of the semi-dwarf varieties of wheat which are called Mexican wheat. Mexican wheat. Okay, this Mexican wheat was having some peculiar characteristics. First is resistant to lodging, and it was having better yield, and it was resistant to the abiotic stress. Abiotic stress like temperature, etc. And it was having a short growing period. Short growing period. So, having these many characteristics, these Mexican varieties have a high yield. Okay, so when this variety crossed with Indian wheat varieties, there are two main varieties developed in India. They are Sonalika and Kalandsona. Remember, these are the two varieties of wheat developed in India by crossing the Indian wheat with the Mexican wheat varieties. And remember, they are the semi-dwarf. It is tall, it is dwarf, and this one will be semi-dwarf. Semi-dwarf simply means medium height. It is neither too tall, it is neither too dwarf, but it is a medium sized crop plant. Okay, because this having less height of the plant, it will be resistant to lodging. Means when the fruit and the seed is of more weight, the plant may fall down. If it is of more height, but when height is reduced, it can tolerate the weight of the fruit and seed. Okay, that's how the varieties of wheat like Sonalika, Kalandsona have brought a drastic increase in wheat production in India. Okay, so these are some of the details about the wheat as one of the high yielding varieties of crops in India. Okay. we shall see about rice, sugarcane and some of the millets in the next class. Okay, till that keep learning, and if you have got any more doubts, please do ask in the WhatsApp group. Okay, of course we shall come with your answer. Right, till that stay home, stay safe. See you in the next class. Thank you.